Welcome everyone to Ecology Live. I'm Jane Hill, Chair of the Publications Committee at the British Ecological Society. And thanks again to NHBS who are generously sponsoring today's talk. They supply equipment and books for ecologists and conservation professionals. They're offering free European shipping for Ecology Live viewers until the end of September. And there'll be details at the end of the talk. This week is, of course, the final talk in this series of Ecology Live. We started this series back in April, right at the start of the coronavirus lockdown in many countries, and have been thrilled to have over 5,000 people register to watch the talks live on Zoom, and have a further 18,000 views of the videos on YouTube so far. And because of its amazing success, we've decided to bring back Ecology Live for another series next year, with more great speakers and more fascinating ecology. And we're already looking forward to it and hope you are too. The best way to hear about these future talks is to subscribe to our newsletter at www.britishecologicalsociety.org/subscribe, even if you're signed up to this series of talks on Zoom. In a moment, I'll hand over to our speaker, Douglas Yu, one of the associate editors for the BES journal Methods in Ecology and Evolution, who's giving today's seminar on how environmental DNA can be used to measure and map biodiversity. So please do use the Q&A box to submit your questions during the talk. There's no need to wait until the end of the talk, and they can be named or anonymous, you can select. So Douglas, over to you.
Okay. I don't see my keynote file. Here, does that work? Okay. Someone tell me if this is working.
It's good. That's fabulous, Doug.
Okay, thank you very much. All right, well, my initial hope was that I would have no screw-ups, but already managed to do that. So thank you very much for the invitation. I've been following the Ecology Live talks myself with great interest, and so this is the last one. And what I'm going to present today is mostly unpublished, which means the results are subject to change. But the reason I'm choosing this set of results is because I want to give everybody an idea of what where eDNA, I think, is taking ecology, what this new data source will allow us to do. So we'll go for a simple example to start. We'll go to Alaska. In Alaska, there is a wild salmon fishery that's over $500 million a year worth of revenues. And the fish are caught before they enter the freshwater streams to breed. And so there are a few fish that escape. That is the escapement population, or the spawning or breeding population. And of course, you need some fish to escape to make the next generations. And so the question is, how many are escaping? How many are able to do this? This is a really important number. And the state of Alaska actually counts these fish, the escapement. And as long as the number of fish is high enough, they can allow fishing to proceed. But if the number of escaped fish is too low, then they start to close rivers temporarily. So you can see this, they issue an announcement every week. Here's one from last week. This closing a couple of rivers temporarily and so on. And so this is a nicely managed fishery. The challenge, though, is that there's like 1,000 streams in just this part of Alaska, Southeast Alaska, used for spawning, but they only have about 14 counting stations. And so a very large fishery is being managed by data from a very small number of counting stations, because it is extremely difficult to set up these counting stations and to put personnel in them. The best one of these is the Elk Creek Station in Juneau, and you can see it's an all day, every day affair for many days during the entire summer. And so we went there to see if we could replace or substitute or augment or scale up what they do using eDNA. And so they count species. They also do biomass weighing and sexing and things like that. All we wanted to do is see what we can do is count. So they have many people all day, and we had one of them sample some water at the end of the day, once a day, put the filter into the freezer, and then eventually take all the samples and measure the DNA using qPCR, quantitative PCR, which measures the concentration of DNA per species. And so let's look at the results. Count data, these are their count data. You can see that the fish go up and down over time. The red line is total fish. And here are the eDNA results. And they are terrible, it looks like anyways, because, well, not so terrible, right? You have, when you don't have fish, they don't have DNA. When you do have fish, you do have DNA, but you're not really getting those little peaks and troughs. So what's going on? So what's going on is that qPCR gives you concentration of DNA, but what you really want is a volume, mass, right? And so you also have to take into account water, the volume of water, the flux of water. And because this is America, they count flux in cubic feet per second. So we multiply the water flux times the eDNA concentration, and you get, in the end, nanograms per second times the constant. And so that's eDNA rate. And now it actually looks pretty good. When there's a few fish, you get a little bit of DNA. When you have a lot of fish, you get a lot of DNA, and so forth. And the error is mainly driven by error in counting the water flux, not so much the eDNA. And then now you take the eDNA rate and the count, and you see whether or not you can fit a model with one to the other. So can eDNA fit and explain the count data? And it can. Not only that, but the 2015 model and the 2016 model are not significantly different from each other, which means that there's some possibility, right, that we could fit a model to many years of data over many streams, and then hopefully over time get a robust model relating eDNA rate to fish counts. And then this would allow scaling up. So eDNA does contain abundance information, is what I'm trying to argue here. Not only that, but because eDNA sampling is easier, it becomes possible to perhaps set up automatic water sampling stations or citizen science networks with a distribution network, and then get more counts over more streams. Okay, so that's a few species at a time. I'm not really an aquatic ecologist, so I'm much more of a terrestrial ecologist. One of the questions that's really enervated me over the years is to ask, how to measure whether a nature reserve is effective, and especially in tropical situations. And in China and Vietnam, they instituted wildlife trade bans this year because of the epidemic. And so we'd like to know in a few years, is there going to be an upstream effect? Will the poaching actually stop? Will we see wildlife recovery? And to do that, you need to count wildlife. And that's a very, very difficult thing to do. Here's a picture from a Vietnamese rainforest. And there are two large mammals in this photo, which you can't see. But right in the center, there's one. It's a human. It's a member of your own team. So you can't even see your own team members. Never mind wildlife who are trying to stay away from you. So we thought about how we might do this. And we decided to use as our main place is this place called the Ailaoshan Nature Reserve in southwestern China. It's about the size of Singapore, and it's also quite typical. It's the top of a ridge of mountains. And so how do you measure wildlife? What we want to do is we want to know what species are there, and then where they are, where each one is in this gigantic park. Okay, so we decided to use leeches. And leeches, they feed on blood, they fill with blood, they go bury themselves somewhere, they digest the food, and when they go out for the next feed, they have some residual blood still in their bodies, and that you can take that blood, that residual blood, amplify it, and read the DNA, and get an idea of what species was in that leech. And you can do this en masse. And leeches are sit and wait predators, so they will feed on lots of different things, from frogs all the way up to bears. And the way we did this was we issued collecting tubes with RNA later to 160 or so park rangers, and they did their patrols, and they ended up collecting about 30,000 leeches in 2 months. And then our lab just then choked on it for a while. And then we amplified up 12S gene and the 16S gene, and you can see we get a whole range of vertebrates, from ground-dwelling birds, perching birds, frogs, and large and small mammals. Okay, so a couple of pieces or aspects of the pipeline are quite important. What we were able to do is we were able to force the rangers to do replicated sampling for occupancy modeling by giving them small tubes, and as they filled one in, then they would fill the next one and the next one. So in a single patrol, they were able to take essentially replicate observations in portions of their trail. And so that made things a lot easier. We didn't even have to train people to do that. It just happened naturally. There was a very long molecular and bioinformatic pipeline. It took us a long time to figure out. And our bias is to minimize, is to reduce false positives as much as possible. And one of the pieces that we did to do this was we used a statistical wrapping software called PROTAX, which assigns taxonomy to these very short DNA fragments with a probability estimate. Finally, we take these data tables and made a multi-species occupancy model. And so let me go over that very quickly. Occupancy models separate some ecological process from the process of which you observe that ecology, right? So you're in a place and a species is either there or not there, okay? And so there's some probability that it is there or not there. And in our case, we chose two variables, elevation of that site and the distance to reserve edge as variables to model the process about whether or not a species is present. Now, once it is present, if it is present, then there's a probability that you will detect it or not detect it. And that depends on lots of things. And in our case, we were able to measure the number of leeches per tube, right? And then that gave us, so that's like effort, sampling effort. And then we use that as a partly to parameterize the detection probability. Okay, so some quick results. Here's the map of the park, and the light color means that we modeled probability is higher, and the muntjac is found in lower elevation sites and also towards the edge, which makes sense. There's a leopard cat, and this is the opposite. This is a cat this species is found more often in the middle of the park and the core of the park, the less accessible sites. And if you take all those maps and add them together, you see that there's more species in richness in these higher elevation, less accessible sites. But we don't want to know just how many species, you want to know which species. So we took every site and we made got the composition of species that were modeled there, and then we ran a principal components analysis, and 50% of the variations explained by principal components 1, and principal components 1 is distance to edge of park, basically, or elevation. So it's a site accessibility. So now we can ask, for every species, where is it on PC1? And so we can see that, for example, there's a bunch of species in the mammals that are found towards the outside of the park. And that includes 3 domesticated species that people bring in to forage on the edge of the park. So that makes sense. Most of the mammals, however, are not on the edge. They're found in the core. They have a positive PC1 value, and that makes sense. As well, most of the avifauna are found towards the core of the park. Again, that makes sense. And there is hunting of birds in these parks. And then finally, the amphibians, maybe about half are found towards the core. So there really isn't a sort of core edge difference in amphibians. And that kind of makes sense too, because amphibians, maybe they're not hunted. Maybe their distribution is determined by things like microtopography or something like that. So the overall, we were able to use leeches to map wildlife across the whole park. We were able to get the data, the raw data, in about 2 months, and then took a while longer to get through to process it in the lab. But it's something that you can repeat in the wild pretty easily. We found that everything from Asiatic black bear to birds, and most of the large and small mammals and so on were found to the core, and then most of the small mammals and domesticated animals were found outside. So this becomes a performance metric that we can potentially use in the future. Okay, so I just want to quickly thank Chris Baker for doing the occupancy modeling, Ecology for the pipeline, and Wenqing Zhong from the Yunnan Forestry Institute, who heard about this leech thing and asked us if we wanted to do this. So it comes from them. Now, that was a tropical forest. What happens if you go to England? Now, it's not a tropical forest, and it's, so in this kind of mixed use landscape, there are vertebrates, and there's a lot of semi-natural habitat, but it's an incredibly difficult thing to sample biodiversity here because you can't get access to most places, and there's lots of domesticated animals, and so if you try to use flies, for example, to get DNA, you'd pick up cows and dogs and cats and things like that. So what we did was we took advantage of private sampling, private industry. So in the UK, in England particularly, if you build a house near a pond, you have to survey that pond for this protected species called the great crested newt. And the way it's done now is that the home builders pay a specialist company, they send a professional ecologist out, they sample some water, send it to a lab, and then that lab then tests for presence or absence of great crested newts, qPCR. And on top of that, Nature Space is a private company, and they partnered with Nature Metrics to sample systematically 500 ponds across the South Midlands of England. And then they took that result and made a single species great crested newt distribution map. And so you can see areas red and amber and green, which are modeled to have a higher prevalence of great crested newt. And now you can just pay some money depending on where you are in the map, and then that money then goes to not just surveys anymore, but to building 4 more ponds. So that's a biodiversity offset thing. And those samples were sitting in freezers, and we thought, can we metabarcode them? Could you give them to us, right? And so they gave us them, and there was DNA left over for about 300 or so, and we managed to get metabarcoding data out of these things. So basically, we got a couple $100,000 worth of samples for free. And my PhD student, Taiwan, ran through the metabarcoding pipeline, got about 76 species of vertebrates, of which 47 are found in 5 ponds or more, so we can model them, including the great crested newt. And we use a package called our SJSDM, which is a joint species distribution modeling package. This is in beta. And what we did was we asked this mammal community in each pond, right, as detected by eDNA, is it explained by the pond variables? So these are professional ecologists, like pond area, mapping these things. Or can we use land cover around each pond? What's the dominant habitat around each pond, plus the spatial location? So it's a pretty straightforward model. And I'll just show you some very quick results. In the 5 amphibians, the smooth newt and great crested newt are found more often than by chance in smaller ponds, which we know is true. They're also found in ponds that are more likely to be found in ponds with great macrophyte cover, and they use macrophytes to lay their eggs. The other species don't seem to be affected by these things, but the surrounding habitat around those ponds does seem to affect their prevalence. So they tend to be found in places with more woodland, and that's true for the palmate newt, interestingly, which is known to be like cooler temperature. And we could do this for all the species times all the variables, but I won't do that to you. Okay, so what I will do is show you a kind of summary of the results. And it's starting to look like, to me anyways, that we're getting something that Matthew Label and all are calling the internal structure of metacommunities. So you can map that here in a ternary diagram. Every point here is a species. And if you're towards the lower left, these are some species, which happen to be the fish species in our data set, whose distribution is almost perfectly explained by the variables that we happen to have. Then there's another set of species whose presence or absence in these ponds is not well explained by the pond variables or by the habitat. But if they're in one pond, they tend to be found in a pond nearby, spatial autocorrelation, even if those ponds and habitats are different from each other. Those happen to be mammals and birds that kind of make sense. They're terrestrial and they're moving back and forth over the landscape. And then there's a bunch of species that we're not able to explain very well. And that's probably because we're missing some environmental covariates. But overall, if you look at it, most of the points are towards the lower left, and which means that we seem to be doing a halfway decent job of explaining distributions by environmental covariates. So more towards the filtering end of the metacommunity structure. Okay, so that's where we are with this. Let's end with one last question, which is not really an applied question. How will biodiversity respond to climate change? Or it's like the biggest applied question of all. And the normal way, the most frequent way that this question is approached is by, you have some climate current, you have some modeled future climate, and then you have your species and its climate envelope or distribution, and you move its climate envelope north or up and so on, following the modeled climate. But everybody knows, and I'm not a climate change college, but everybody knows that that approach, the correlative approach, deliberately ignores the fact that species interact with each other, because that information really isn't available. So is it possible to get that kind of information out of data, even if you don't have direct knowledge of species interactions? And I was really fortunate to be invited to work with a group of Finnish and Danish ecologists, and they have the set of samples from Greenland, which is up in the Arctic Circle. And you go way up there, and it's basically an ecosystem of flies and spiders and wasps and so on. And they've been catching these flies and spiders and wasps using pitfall traps and pan traps for the last 20 years or so, from 1996 till now. And they've been sitting in there in alcohol, they've been sorted and so on, but we were able to reconstitute the trap samples and then pull the DNA out of each of the trap samples and then just send it to shotgun sequence. So we just sequenced all the DNA in every trap sample. That's 492 sequencing libraries plus a bunch of replicates. So we ended up sending something like 750 sequencing libraries to sample, and there was terabytes of data sitting on Amazon S3. And we also assembled mitochondrial genomes for most of the species. And what you do is you take the trap samples reads and you map them to all the mitochondrial genomes. And then sometimes they map, well, so here's one where that species has attracted a lot of reads. So we say that species is present in fairly high biomass, right? And here's a species that has attracted a lot of reads, but we rule it out because it's only mapped to a small part of the genome. So it's probably a conserved site. So we can separate true positives from false positives, which is really important in this kind of work. That pipeline is published in molecular ecology resources. And what you get in the end is a species by trap sample table. And so what I'm showing you here is just a subset of those data. And I call this a community time series. So on the left is 1997 to 2013. And the first thing you should notice is that summer is now twice as long as it was 1997. So that's the climate change effect. And that's pretty much all that's happening in this place. So we're abstracting out only climate change on this this community. And this is being worked up for publication. There's a couple of summary statistics. First of all, the temperature has increased by about 2 degrees during this time. And what was a surprise to me is that the number of detected species, the number of observed species in our samples dropped by 50% or so during this time, which really is a surprise to me. So the question is, well, what's going on? It's not like there's toxins up there unless you count carbon dioxide. Well, it's really difficult to read, right? Because if you see that one species with the blue square around it, that means that red means that species had a biomass peak, an activity peak sometime in August 1997. And if you go to 2013, it doesn't, right? That makes sense because it's warmer earlier in 2013. So as it happens, we do see a biomass peak earlier in 2013, in July. Okay, that makes sense. But it's very rare to see something as simple as that. Here are 3 more species, biomass peaks in August, and nothing in August, July, or June. In fact, there's a whole bunch of species that you see that had this phenological behavior of having a biomass peak sometime in 1997 and nothing when you get to 2013. And vice versa, there's a whole bunch of species that didn't show these biomass peaks, that didn't have that kind of activity, but when you get to 2013, there they are, showing biomass peaks. So the phenological activity has changed. What on earth is driving all this? It's not as simple as just it's getting warmer, be active earlier. Okay, so we're really fortunate because we're working with Nerea Abrego and Otto Vizcaíno, who literally wrote the book on joint species distribution modeling. I urge everybody to read this book. What they did was they built a model where the response variable is each species present, absent in every trap week for a year, right? And then you ask, is it present or absent based on last month's temperature for that week number, the week number squared to model this kind of phenology, and then take all the incidences of that species from last year and add them all up, and that's last year's abundance, right? So for intraspecific density dependence. And then you take all the herbivores in your data set from last year, add them all their incidences up, and that's last year's herbivore abundance, and last year's parasitoid abundance, last year's predators abundance, last year's detritivore abundance. So that's trying to model in the simplest way possible interspecific interactions on an annual basis. And you're doing this simultaneously, jointly for 52 species. And what you do is you can model with the simple model with just temperature and week versus the model with species interactions. We can see which model performs better. And when we ask what model performs better, we're actually asking 2 questions. So the first question is, which model fits better to all the data? And there's a measure of that power, and if it's positive, then it's fitting to the data better than random, and if it's negative, it's fitting to the data worse than random. And that simple model with just temperature and week fits better than random to all but 4 species, and it's worse than random for 4 species, even though you're fitting to the whole data set. The interactions model, the complicated model, fits better than random for all the species, and in fact fits better as in general. Okay, so you could say, well, it fits better, it has more terms. That's true. So now we can ask the second question: predictive power. How well does these models predict a missing year? So you fit the model to 14 years, you hold 1 year out and you ask how well does it fit to the year that you held out. And so in general, the prediction is worse. It makes sense. You're fitting to less. You're trying to do something more difficult. But the abiotic model does really badly, right? It's temperature and weak, and it predicts better than random for 20% of the species. 80% of the species are fit worse than random or just random. And the interaction model is better than random for about 50% of the species. So what we conclude, right, is that the trophic interactions model does a better job of explaining that fitting and predicting the prevalence of species. So we infer, right, that trophic interactions, particularly trophic cascades, are having an effect on climate change response, or the biodiversity response. So I don't have a great answer about how we're going to predict how biodiversity will respond to climate change, but I think these community time series, which are going to be more and more feasible to do, will help us get insights that have been difficult to get until now. Okay, so to very quickly sum up, these are some questions that I went over, and what I'm just trying to show you is that a lot of different technologies are coming together, plus field work, to allow us to generate these high-dimensional data sets much more easily than has ever been possible before, and then new statistical methods are allowing us to build much better predictive and explanatory models of maps than we've ever been doing before. And I think this transition to higher technology and so on is also transitioning our science from a natural science, a basic science, into an engineering discipline, right? And if you're an engineer, you're interested in the science, but you're also interested in quality control, the use and invention of instrumentation, standardization of protocols, automating processes as much as possible, and most importantly, scaling up production of data in our case at high quality and low cost. And I think that is kind of where I think ecology is going because of eDNA and statistics and so on. And it's going to change the way we train, the way we reward, the way we organize ecology. Okay, so thank you very much.
That's great. Thank you ever so much for that talk, Douglas. So we're going to go straight to questions on this fascinating subject. So we've got questions coming in on all of the different components that you talked about. So I'm going to start off with a couple of questions about leeches. And as somebody who works in the tropics, it's great to see leeches being put to good use, especially leeches that are no more. I'm delighted to hear about that. Anyway, there's a question from Monica Kaushik, who asks, I understand that the pheasants or the ground-dwelling birds would have been been eaten by the leeches, but she's wondering about the passerines, and just wondering about whether your objective was not necessarily to sample every bird species. I guess it's sort of a question about how well the leeches sample the biodiversity that is there.
So the ground-dwelling birds are sampled better, not surprisingly. We get more detections of them. We were surprised we got any passerines, and they do get them. And these are leeches that are picked up off the ground, right? So somehow they're getting some birds. I guess they must. Some of them are occasionally hit in the ground. Some of these leeches might also be arboreal. We don't really know. We in other data sets we've gotten even things like gibbons, bats, right? So leeches seem to get around.
And a related question about using the data to make your predictions for presence of species. Catherine Stewart says, how much does dispersal ability play a role in your occupancy modelling? Is it a parameter that you include?
No, mainly what we're worried about is, of course, whether or not we're violating the closure assumption. So there's two ways you can violate the closure assumption. One is that the animal leaves, right? And so by the time you actually pick up the leech, it's gone off somewhere. So the answer is, you have to set a large enough patrol area. So we don't have a huge amount of control over that. But all the sampling was more or less done around the same time. So it seems kind of unlikely we're picking up the same animal in two different patrol areas. But it's still possible, but unlikely. The other one is whether or not the leech travels. There are scenarios, but they seem really rare. Like they have to feed and then ride on an animal and then go somewhere else and so on. But once they come off, I mean, they're crawling and going into the ground. So the leech side probably doesn't violate the closure assumption.
And yeah, I'm going to move on to ask you a question about some of the pond data. So there's a question here from Dr. Snedden, who says, wouldn't it be interesting perhaps use these techniques to look at the rewilding project at NEP, I guess, thinking about picking up species that arrive or are in as a consequence of the rewilding project, perhaps before you can see them easily by other methods?
Yeah, especially for small mammals and so on. I mean, technically, because I'm inclined towards that, I want to convert from qPCR or PCR to this CRISPR-Cas method called Sherlock and Carmen to pick up really, really rare things. But right now, I mean, I'd love to do this, so call me. But the issue primarily is I'm leaning towards situations where there's lots of standardized sampling already taking place, already financed, and then taking advantage of that.
And then there's a question, anonymous question, asking about the Arctic work. Just asking how easy it was and how successful you were in amplifying DNA from the 1997 samples compared with the more recent samples, and just a question, I guess, about the methodological challenges with working with old material.
So it's all been held in alcohol the whole time, and we didn't amplify. What we did is we just sequenced, we shotgun sequenced. So you take all the DNA, fragment it, build a library, and send it, and sequence it. I have to think about. I mean, I didn't see any real difference in the mapping and so on. And note that we also got twice as many species detections in 1997 as we did in 2013, right? So if anything, it's if it's a degradation, it should work the other way around. But I have to think about, like you'd have to do things like measure the fragment of DNA. We went for quite small insert size to make it possible.
Okay, and then the final question, another anonymous question, a more general question about the future of eDNA techniques in poorly studied parts of the world where maybe there aren't any species DNA barcodes in existence. Just a more general question about how you see the benefits of eDNA being applied in other parts of the world with less information, but perhaps potentially very high biodiversity.
So I mean, we do a lot of our work in China, as you saw, and it's actually, even the mammals, and it can be difficult to get species. So actually, one of the issues is that you have to that's why I use ProText, because it allows you to model the possibility that there's a missing sequence in your data set, and then gives you a probability of the sequence coming from your read coming from some missing species. You can get higher level taxonomies from things. So when we work with insects from places like southwestern China, we mostly modeling things by where they place on the phylogeny, some skeleton phylogeny, rather than trying to get a species identification. And that seems to produce a lot of useful information. So those are the kind of approaches that we use.
Brilliant. Thanks ever so much, Doug. Okay, I'm going to share my slides. Okay, so thanks ever so much, Douglas, for your talk, and that brings Ecology Live to a close. And if you've missed any of the talks, do remember that they're all up on our YouTube channel for you to watch at any time. And once again, if you want to keep up to date with our news and activities, please do subscribe to our newsletter. We'd also love to know what you thought of the series, so please look out for our feedback form in the next couple of days to tell us how we can improve for 2021. And to end, we want to say a huge thank you to all of the amazing speakers for taking part in this series. And a big thank you to our steering group, which alongside myself has included Mark Cadott and Pete Manning, and a massive thank you to all of the BES staff involved too. And finally, we'd like to say a huge thanks to you all for joining us in such fantastic numbers week by week. And I hope you agree it's been a delight to have this weekly dose of the latest and best ecological research during this time of upheaval. So I'll leave you with details of the offer from the sponsors of today's Ecology Live talk, NHBS, so you can take full advantage of the offer. So take care everyone, and we hope to see you all again soon.